In this video, we will discuss how to reduce dimensions using principal component analysis. Similar to single-cell RNA-seq analysis, we run dimension reduction before clustering to simplify the data and making the clustering easy. First, we determine the significant principal components, and then we use them to cluster cells with graph-based clustering. The dimensions have to be reduced because the gene expression values of each gene measured can be thought as the dimensions to data. As we can have thousands of genes, we would also have thousands of dimensions. This would make the clustering complex and visualization of the clusters impossible. But often the gene expressions have correlations, and with dimensional reduction we can remove these redundancies in the data. This way we can have lower number of dimensions but still have the most relevant information of the original data. We're using principal component analysis PCA for the dimension reduction. It is the linear dimension reduction method which finds principal components that explain the variance in the data. These components are computed so that the first component accounts for the largest possible variance in the data, second component the second most, and so on. After this, the most important PCs are selected and used in the clustering. The variance can be viewed as the information in the data, so by choosing the most important PCs, we manage to sum on the most important information. This graph here elaborates how by changing the coordinates to another system, we can reduce the dimensions. We're also going to use another dimension reduction method, UMAP, for the visualization of the PCs. UMAP is a nonlinear graph-based dimension reduction method which allows to visualize the high-dimensional graph of clusters in low-dimensional space. In Chipster, you can run the v4 PCA clustering and visualization tool by giving the combined R object as the input. As parameters, you can choose the number of PCs to compute. By default, 30 components are computed. You can also choose to print the PC loadings to a text file. The tool produces the R object that you can give to the next tool. The tool also produces the PDF file containing the visualizations. Here first we have the UMAP plot, which shows the computed clusters. In the same PDF file we also have the clusters visualized on top of the tissue image with spatial dim plot. If you wish to view the spatial location of any individual clusters, you can also run the next tool, v4 Visualize Clusters, for a closer look. You can run the tool by choosing the clusters to view and giving the Seurar object from the PCA tool as input. As output, you get the PDF file containing the spatial dim plot. Here you have the chosen clusters 1 and 2 pictured with the tissue image.